Natalia Elizalde, CBO at VIVEbiotech. We are a company fully specialized on lentiviral vectors. Indeed, we are doing development as well as manufacture under GMP conditions of these kind of vectors, being fully specialized, and therefore, something important to be mentioned is that we have a high degree of virology expertise as well as bioprocess development, which allow us accommodating new programs with a high degree of innovation, but also, of course, manufacturing programs using our plug and play platform in an efficient, cost efficient, and timely manner. 2024 was a key year for us to really establish the larger scale available in the market for the technology we are using. This scale that is being used both for very advanced clinical trials up to commercial as well as for in vivo programs. And this year we have manufactured several batches at this large scale, both in engineering as well as GMP batches. So this is a key fact because this reinforces that we are more and more prepared to cover all the phases up to commercial, always thinking on, of course, reproducibility and cost effectiveness of our process at the end of the day. So this is the key part of this year, as well as the fact that, of course, we are continuously increasing our headcount up to 150 people now. And we continue implementing new capacities in analytical methods development as well as fill and finish automatic capabilities. I'm attending the Advanced Therapies Europe event here in Estoril. This has been a great event in which where I'm having the opportunity to meet different companies. Some of them we are already working with, but also some new programs coming. And here again, what I'm realizing is the importance of the in vivo use of lentiviral vectors. So, of course, different projects in which the vectors are being used ex vivo are being discussed, but now more and more the use of lentiviral vectors as final product is being very, very relevant. As I could mention before, we are involved in 8 programs using the lentiviral vectors in vivo, some of them already approved for clinical trials. So this is allowing us having very relevant discussions on the in vivo use of lentiviral vectors.